In this video, we are going to be looking at the structure of RNA and its function. So before we get started into talking about the structure of RNA, we must first discuss where RNA is synthesized from. And in order to discuss that, we need to talk about the flow of genetic information. The molecule of RNA actually comes from DNA because DNA undergoes the process of transcription in order to synthesize that RNA. RNA is a very important molecule because there are many RNAs that undergo the process of translation to synthesize proteins that are needed in any organism. The figure that we have in this slide represents these processes of transcription and translation, specifically in eukaryotes. In these organisms, in the cells of these organisms, what happens is that because we have separate compartments, we see that the process of transcription and translations are in particular areas of the cell. So in the top left corner of the diagram, we see that that quarter circle structure is the nucleus of the cell, while the white area is going to be the cytoplasm of the cell. As you can see, the process of transcription occurs in the nucleus, while the process of translation is going to be occurring in the cytoplasm. When it comes to the principal kinds of RNA and their structures, the image that we have in this slide represents a single-stranded RNA molecule. And many of the RNAs that are in organisms are going to be single-stranded. Now, when it comes to how we compare the single-stranded RNA to DNA, these are the features that in a way they share with each other. First, the structure of RNA has a 5' terminus and a 3' terminus. Now, the nucleotides that are present in RNA are linked to one another by this area that I'm circling or highlighting in green, which is a phosphodiester bond. And remember that we define that phosphodiester bond as the 3' to 5' phosphodiester bond because of the chemistries that occur in order for it to be synthesized. Now, when it comes to how different is this molecule to DNA, we can pinpoint a few chemical features. First, we know that this is an RNA molecule because at carbon number 2, we have hydroxyl groups. So in this molecule, the sugar motif is ribose. If we look at the nitrogenous bases that are present in this single-stranded DNA molecule, or single-stranded RNA molecule, we can observe that uracil is present. And remember, uracil is the nitrogenous base that is present in RNA and not DNA. I established already that RNA is going to be the template that it is utilized in order to synthesize protein. Now, RNA has many functions, and the molecules that we have here outlined in this table are going to be just RNA molecules. Now, these RNA molecules are what we called non-coding RNAs because they do not transform from RNA to protein. they stay in RNA form, which means that they were made from DNA through the process of transcription. And they stay in RNA form, so the reason why they're called non-coding is because they are not translated to make a protein. Now, they are involved in processes that have to do with protein synthesis. So let's discuss the different types of RNAs that you guys need to be familiar with in this course, starting with transfer RNA. As you can see, transfer RNA is going to be important in the transport of amino acids to sites of protein synthesis. We are also going to be talking about ribosomal RNA, which is the RNA that combines with proteins to form ribosomes that are active. And remember that ribosomes are going to be the machinery that makes those proteins. We're going to be mentioning also messenger RNAs, and messenger RNA is this RNA that I keep on talking about that results from the transcription of DNA, and that's why it directs the amino acid sequence of proteins if that RNA leads to the synthesis of proteins. We are also going to be mentioning about small nuclear RNAs, which are involved in processes of initial mRNA. And this initial mRNA or pre-mRNA is going to undergo modification in order to create a mature version. Understand that this process is particular in eukaryotes. We are going to be looking at interfering RNAs and microRNAs. Understand that those last two RNAs are going to be affecting gene expression, which means that they provide a way in which we can regulate the presence of a particular DNA in the context of the cell. And the difference between the two is that the small interfering RNA, it is utilized by scientists to knock out genes, basically remove some DNA in the context of their experiments. But microRNAs have been shown to be important in growth and development. Let's get started talking about the particular chemical features of each of these RNA molecules, starting with transfer RNA. When it comes to transfer RNA, understand that the abbreviation for it is tRNA. As a reminder, remember that the function is to transport, in other words, bring the amino acids to sites of protein synthesis. Understand that when it comes to tRNA, tRNAs are a single-stranded RNA molecule. The structure that it has, as you can see, that is given by these blue dots that I'm going to also highlight in blue. So tRNAs has these regions of intramolecular base pairing within the single-stranded RNA molecules that results in a structure that is called a hairpin. At times, it is also called a stem loop. But the most important thing, one of the most important things that I want you guys to remember when it comes to tRNAs is that because these are the molecules that are going to be bringing the amino acids during protein synthesis, is where is the location of these amino acids? As you can see, the 3' end, because these RNA molecules run 5' to 3'. The 3' portion of the RNA molecule is where we have the linkage of the amino acid. And the amino acid is linked to the tRNA through an ester functional group. Lastly, we have in these molecules a section that is called an anticodon. And an anticodon is going to be a complementary sequence to the mRNA. And that's through this complementation between the anticodon and the codon that it is on the mRNA is how these tRNA molecules are able to bring the correct amino acids during protein synthesis. When it comes to ribosomal RNA, or rRNA, remember that these are going to be combining with ribosomes in order to be involved in protein synthesis. So the ribosome is actually considered a ribozyme because remember that the ribosome is the machinery that it is involved in protein synthesis directly. When it comes to comparing prokaryotic ribosomes and eukaryotic ribosomes, understand that we have a large subunit or a small subunit. And I'm just going to particularly highlight the large subunits and the small subunits. Now, when we look at how many ribosomal RNAs are in these structures, notice that both small subunits for any of the ribosomes in prokaryotes or eukaryotes contain only one molecule of ribosomal RNA. While if we look at the large subunits, the eukaryotic ribosome has 3 ribosomal RNAs, while the prokaryotic ribosome only has 2. The next type of RNA is going to be messenger RNA. A messenger RNA or mRNA is going to be the molecule that is directly synthesized from transcription. Now, understand that when it comes to prokaryotes, the process of transcription and translation are going to be occurring simultaneously because these organisms do not have a nucleus. And as I explained previously, when it comes to eukaryotes, the transcription is going to occur in the nucleus, and the mRNA is going to be processed and then exported from the nucleus before it's involved in the process of translation. So, in eukaryotic cells, in eukaryotes, when we have this mRNA that is produced from transcription, okay, it's made as what is called the pre-mRNA. It is to be said that this pre-mRNA has to turn into a mature version of mRNA. A small nuclear RNA or snRNA, understand that are going to be involved in the process of splicing, specifically intron removal, okay, from that first mRNA that came from transcription. And when that occurs, the removal of these introns or non-coding regions, what is going to be happening is that we are going to have a mature RNA that has only exons. Next, we are going to be looking at the 2 different types of RNA that are involved in mediating gene expression, starting with small interfering RNA or siRNA. So understand that when we have an siRNA, they are going to be eliminating the expression of an undesirable gene. This is the one that it is utilized by scientists to just take off or knock down some genes in a particular experiment. Understand that when it comes to siRNA, they start as a double-stranded RNA molecule. Dicer is an enzyme that is involved in cleaving that RNA into smaller sections. And then in the case of siRNA, these small sections are going to be binding their mRNA, and that mRNA that it is the target is going to be sent to be destroyed, and that's why we don't see the production of that particular protein. So this type of gene regulation is by not allowing the mRNA to ever reach protein, meaning the target mRNA. So this is going to be the mRNA of interest. When it comes to microRNAs or miRNA, understand that these start as a single-stranded RNA molecule that also this enzyme dicer acts on it, and it creates the microRNA. Now, even though there's many mechanisms, one of the ones that is well studied is that these microRNAs are going to be binding to the mRNA of interest. Meaning, mRNA of interest means what you're interested in studying. And in this case, the mechanism that it is executed by that microRNA is that it's going to be blocking translation by creating, as you can see, a hairpin-like structure.